All right, let's move along to our next lecture on genetic changes and genome evolution. So remember your learning objectives and key terms, and let's dive right into it. So mutations are any heritable changes in the DNA, meaning the code of the DNA, the order of the A's, T's, C's, and G's, something about it gets changed. A letter gets swapped, things get deleted, things get added. And then this mutation gets passed on to the next cell or to the next organism if you are a sexually reproducing organism. So let's discuss the different forms of mutations. There are point mutations, and this just refers to any kind of change in a single base. There's insertion and deletion, where we add or remove one or more bases. A lot of these can also be extrapolated to discuss the whole chromosome. We can add or remove entire pieces of the chromosome that can include one or more genes. We can cause inversions, so part of the code will flip and go upside down. So the DNA is in the opposite orientation. We can duplicate a gene where a certain number of codons or bases get duplicated, and so we see the same amino acids or something like that occurring. In or in transposition, this is where we have a transposon or a mobile genetic element that jumps around within the genome and moves its location. Sometimes this can be into the middle of a gene, or when it jumps away, it may take a portion of a gene with it. And then there is reversion, where the DNA can mutate back to its original sequence. Now, mutations can fall under a couple different classes. These were mainly talking about just nucleotide based mutations, not on the chromosomal level. So there can be a silent mutation. This is where something like AAG gets mutated to AAA, and this still codes for the amino acid lysine. So there's no change in the overall sequence of amino acids. It's silent. We can barely tell that it's there unless you go deeper and look at the code that it came from. Then there are missense mutations. This is where the amino acid that was originally there gets substituted for a different one. So this is the case of AAG being mutated to AGG or ACG, so an arginine or a threonine. Sometimes this does not really cause too much of a change in your overall protein structure. If you are converting the amino acid to one with a very similar chemical structure overall, like a hydrophilic to a hydrophilic, and they're roughly the same size, maybe your protein won't really have any differences at all. But if you convert a hydrophilic amino acid to suddenly a hydrophobic one, maybe you're going to start seeing larger changes in the overall protein so that the structure changes and therefore the function will change. And then there are nonsense mutations. This is where the amino acid gets converted to a stop codon. So following our same example here, this is where AAG gets converted to something like UAG, where now there is no amino acid or tRNA corresponding, and we halt translation early. And then there are frame shift mutations. These are usually terms that we give to insertion or deletion ones because they're changing the reading frame of your gene. Remember, we have three letters together making a codon. Well, if I were to accidentally add a letter in between one of my three codon letters or delete one or more letters, I am then going to shift my whole reading frame down or up, and depending on whether it's an insertion or a deletion, and then every single amino acid after that point has been changed because we are shifting the reading frame. So how do mutations arise? They are usually spontaneous changes, and they are rare because DNA has many different proofreading and repair mechanisms that we will talk about. But DNA polymerase 3, if you remember from our discussion of replication, the book portrayed DNA polymerase as a ring, an open ring. And it's due to the shape of the polymerase that it has its proofreading ability. Remember that the purines have a double ring structure, whereas the pyrimidines have a single ring structure, and usually a pyrimidine bonds to a purine. Well, if we had an instance where a purine bound to another purine, a double ring to a double ring structure, this would cause the overall shape of the DNA to be slightly larger than normal, and the overall double strand would bulge, or two pyrimidines together, it would collapse in on itself. And so DNA polymerase, as it is scanning and adding nucleotides, if it runs into one of these bulge areas or areas where it's collapsed, it no longer fits the polymerase properly. And there it can realize that there is a mistake in the code at that point because it no longer fits. So what are the four mechanisms that can cause mutations to arise? The first one is tautomeric shifts in the DNA bases. And basically, what this is some type of spontaneous change to the chemical structure. So, if we see here the adenine and the thymine, this is their normal base pairing. You can have an instance where this bond between the nitrogen and the hydrogen is suddenly removed, and now they are experiencing a hydrogen bond, or the bond is transferred, um, or they're now experiencing a hydrogen bond rather than a normal covalent bond. This results in the thymine being able to pair now with a guanine instead of an adenine. So instead of forming two of those hydrogen bonds, it can now form three, and it will mispair with the wrong base. So it's things like that. Now, the second way is through oxidative deamination. So, this is replacing an amino group on the base with a keto group, this double bond to the oxygen. And this is usually due to some type of ROS, or oxidative stress. And so, this is bad because it'll take something like a cytosine and suddenly turn it into a uracil, which is a completely different base structure that's not even used in DNA. So, any type of instance where we have a change like this, where it causes a or an improper or a mistake in the base pairing, this is just the consequence of it. So, if we have A and T, and the T is shifted to its enol form, then the strand with the A is going to be copied just fine. It's going to be an A and a T, and therefore we're going to keep the same wild type, or the correct orientation of that code. But then in the bottom strand where we had that shift with our thymine, the thymine is now going to accidentally pair with a guanine. And that alone creates a problem. But then when we look at the consequences of when we mutate that, one will still produce the wild type shape from the T, but the other now has a G. So we've converted an AT in the sequence to a CG in the sequence. The third mechanism is the formation of apurinic sites. Remember that A means not or against. So this is a not purine anymore. What this means is, with our A and G bases, you have the phosphate group, the sugar, and the base. In these events, the actual base portion of the purine gets removed. So you're just left with the backbone pieces, the phosphate and the sugar, with the base removed. So you can see the bases still remain on these outer nucleotides, but not on the inner nucleotide. Now that creates a gap in the sequence, and it's going to cause disruption in transcription or translation. We're going to have the polymerases stall because they don't see anything there to add, or it can actually end up creating breaks in the strands of DNA. The fourth mechanism is damage caused by reactive oxygen species. So that can cause all of the previous changes that we've seen. It can induce single or double stranded breaks. It can cause oxidation of the pyrimidines. or it can cross-link the DNA. So now, instead of the bases being bonded to each other by hydrogen bonds, which are readily broken, they are now linked by covalent bonds, which take significantly more energy to break through. Now, a mutagen is any chemical or environmental condition that can induce a mutation in DNA. There are chemicals that can do this. Um, One example of an intercalator which forces a frame shift is ethidium bromide. If you've ever done gel electrophoresis, this is the chemical that you add that eventually causes the DNA to fluoresce. What it does is it fits in between the bases, and that's what causes the fluorescence. Well, DNA in a gel, DNA in bacteria, DNA in us is all the same. So if you were to get ethinium bromide on your hands and it gets into your cells, it can induce these frame shift mutations as well. Then there's electromagnetic radiation. We all know that things like X rays and gamma rays can cause mutations. Well, they do that by forming breaks in the DNA. UV rays, on the other hand, form what are called pyrimidine dimers. So if we're looking at the DNA, here's the backbone, here is the base that corresponds to that nucleotide, and then here is the hydrogen bonding base pairing. So this is another base, and they're just missing the backbone on this side. So I know it's a little confusing. But in these cases, when there are two pyrimidines next to each other, like T's, they can end up forming these cyclobutane rings where they actually cyclize with each other rather than the bond forming between them and their corresponding base pair. And this can cause bulges in the DNA and cause breaks in the DNA, and overall leads to an inability to replicate this portion correctly. Now, if there is a mutation, it's very important that the cell be able to fix this mutation. So we have two mechanisms of DNA repair, two categories of mechanisms of DNA repair: error proof pathways and error prone pathways. For the error proof pathways, these are going to actively prevent mutations, and there's a bunch of them that we're about to talk about now. For error prone pathways, these ones risk introducing mutations. They are kind of like last resort solutions. The cell is already highly mutated, there's significant DNA damage, and we're going to initiate this emergency kind of like save the cell sequence that could create more mutations, but in the end, if it keeps the cell alive, it was worth it. So let's talk about the first error-proof pathway, and that is methyl mismatch. When we are um, proofreading the DNA and we find an error, how does the cell know which strand was the template strand and which strand was the new strand? Which one had the mutation occur in it? Well, when we are doing this, the cell uses methylation to be able to tell the difference. Remember that bacteria will methylate their DNA once it is made, but that takes a refractory period. So in uh, DNA replication, seq-A will bind to DNA where half of it is methylated and the other half is not. And that kind of creates a waiting period before we can initiate a new round of replication. It takes time for the enzymes to come around and methylate the new strand of DNA. And remember that DNA replication is semi-conservative. So the new DNA that is formed will always have one strand that is old DNA and one strand that is new. Therefore, one strand will be methylated after replication, and the second strand will not be. So this is how methyl mismatch works. The parental strand is going to be able to we're going to be able to tell the parental strand and the new strand apart because of that methylation, and then the cell will selectively remove the unmethylated mismatch and replace it with the correct base using the methylated strand, the parental strand, as the template. So let's jump into this. There is a protein, mut S. It comes in and binds and recognizes the mismatch. So it can see where the DNA is not in the correct shape and it will bind there. Once it binds, it recruits two proteins, mut L and mut H. So mut L is going to bind near the mismatch, not on the mismatch. We already have mut S bound there. Mut H then comes in and associates with mut L so it knows where to do its work. So mut L is the guide, mut H is going to do the work, and its work is that of an endonuclease. These are enzymes that cut DNA. So it will create a cut in the unmethylated strand. So this is a single stranded break in our DNA, and it's going to be upstream of where that mismatch occurred. Now, the exonuclease activity is going to remove the damaged DNA, and then DNA polymerase 1 comes in and fills in the gap. So it's going to remove DNA past the mismatch and create a gap here in our new strand of DNA that has to be filled in by DNA polymerase 1. Now, the question then comes, what happens if the methyl group is added before mismatch repair can occur? Because this is all contingent on the fact that we are catching it very shortly after the strand is replicated. What happens if the cell doesn't catch it fast enough and the enzymes come and methylate the new strand of DNA? Well, in that case, we have now incurred a permanent mutation that will be passed down in subsequent rounds of replication. So it's kind of like catch it while you can, otherwise, it becomes incorporated into the genome. There is a video on this. I will post that for you guys as well. And definitely watch that if my explanation was not thorough enough for you. The next way that we can do error-proof repair is to repair those dimers. So when we form that cyclobutane ring between pyrimidine dimers, there's two ways that we can fix this. The first one is photo reactivation. So the enzyme photolyase will bind to the pyrimidine dimer and cleave the cyclobutane ring. But photolyase requires UV light in order to work. This makes a lot of sense. We don't want photolyase using up energy and looking around and trying to fix any kind of problem that only exists if sunlight and UV light is present. So in the dark, photolyase won't work because there's no UV to induce these pyrimidine dimers. But if UV light is present, there is more likely an opportunity for these rings to form, resulting in the photolyase being needed to cleave the ring. Then the next one is nucleotide excision repair. So this is just an endonuclease enzyme that will come in and cut the DNA, and it'll cut it on a single stranded cut rather than a double stranded cut going through the whole DNA molecule, and remove certain types of damaged bases, one of which is these dimers. And then it will repair them with DNA polymerase. So there's a video on that as well. I'm not going to go over the details of that on the exam, but it's good to understand these because it will help you learn how these mechanisms work on a broader scale, which will allow you to pick up on new things that you learn in the future. The next error-proof pathway is base excision repair. So this requires enzymes called DNA glycosylases. They are going to recognize specific damaged bases and remove them without actually breaking the phosphodiester bond. So they come in and they just remove the nucleotide or the nitrogen base portion of the nucleotide, leaving the backbone intact. So we're missing a little base right there. What this creates is an A-basic, a not base site, or an AP site, purinic or apyrimidinic site. Now, this AP site is recognized by endonucleases that can come in and cleave those phosphodiester bonds in the backbone. Once they do that, the A-basic nucleotide is removed. And this allows DNA polymerase to come in and fix that missing aspect of the DNA. So first we remove the damaged base, then another enzyme comes in and says, hey, there's no base here. Let's cut this portion out so DNA polymerase can come in and remake that site with an undamaged base. And there's a video for that as well. Then we have error-proof repair pathway. I believe this is our final one, recombinational repair. So this is going to take place at the replication fork in the event of a double-stranded break. So this is all predicated on the fact that our homologous regions of the chromosome are still very close to each other. And we're going to use the one chromosome that does not have the DNA damage as a template for the chromosome that does have the DNA damage. So this is particular for double-stranded DNA breaks, where the break is actually completely separating the DNA from itself. So the strands will get cut back, and then we will use the intact sister chromosome as the template, and we'll create this really interesting three-stranded hybrid, and that will help us fill in the gap. So let's go into that in just a little bit more detail here. So Rec-A is a protein that we're going to talk about a bit more today. Rec-A binds single-stranded DNA, and Rec-A has a couple of different functions that it can serve, but it all starts with saying, hey, this DNA is single-stranded. That's not supposed to happen, so something needs to be fixed here. And it will find that single-stranded DNA and bind to it. Then what Rec-A does is something called strand invasion. It's going to come and displace one of the strands from the correct sister chromosome and pull it over to where the damaged DNA is. We are then going to have a really interesting three-stranded DNA molecule here, and we will use DNA polymerase to use one correct strand as a template to fix the broken strand, and that helps us fix the error, and then we just seal the backbones back together. Now let's discuss the error-prone DNA repair. And this is SOS repair, or save our ship repair. Basically, this is saying the ship is going down and we're doing whatever we can possibly try, even if it seems absolutely crazy, in an attempt to save the cell before it dies. So this is induced by extensive DNA damage. Lots and lots of DNA has been broken apart. There's single-stranded DNA flapping around. There's double-stranded breaks. There's single-stranded breaks. Everything is a huge mess. Now, this is going to involve two different proteins, Lex-A and Rec-A. We're going to talk about repressors soon, but just know that a repressor is a protein that will bind to DNA and keep it from being transcribed. So if there's a protein bound to the DNA and blocking the DNA, RNA polymerase can't come in and copy that DNA into mRNA. So it represses or turns off that gene. Now, what Lex-A does, it's a repressor that binds to all of the genes involved in DNA repair. It says DNA polymerase 3 and 1 can already proofread and they can already do some repair. So you know what? Those ones work really, really well. They're part of the error proof pathway, which is better. So we don't want any extra DNA repair enzymes to be turned on at this moment, because a lot of the ones that are in that arsenal waiting to go, they make mistakes. They're not as good as DNA polymerase 3 and 1. So we're going to leave them in reserve and not turn them on unless we really, really need to. Well, when we have extensive DNA damage, you will have a lot of single-stranded DNA that is in existence. And what loves single-stranded DNA? Rec-A. So Rec-A will come along, bind to that single-stranded DNA where the DNA is damaged, and if that Rec-A is near a Lex-A, Rec-A's second ability will come out. It has what's called a coprotease activity. It's going to tell Lex-A to break apart and fall off the DNA. So now Rec-A is just doing its job of binding the single-stranded DNA and trying to fix it, but it also sees that Lex-A is nearby, and Lex-A is its nemesis, and so it says, Lex-A, destroy yourself. Now, that repressor is removed from all of our DNA repair genes. Now, these DNA repair genes are what we call induced, they are turned on. What does that mean? So when this happens, we start creating all these different enzymes that can now come out and help repair the DNA. We already have Rec-A, DNA polymerase. We have all these repair enzymes working, but it's not enough because Rec-A is still being bound to broken DNA. So when Lex-A is removed and we start to make all these new proteins that help in DNA damage, two sloppy polymerases are made. So these are DNA polymerases that didn't quite make the starting team. They're sitting on the bench because they don't, make enough connections. They don't make enough baskets. Whatever it might be, they are not playing the game properly. They make mistakes. So we leave them in reserve. But if everything is falling apart and your star players aren't able to get the job done, then what can it hurt? We're already, losing the game by 100 points. Let's just put in the bench too. So in this case, these sloppy polymerases come in and they will start to fix all of these multiple errors in our DNA. But because they lack proofreading activity, they make a lot of errors, they make a lot of mutations. But the cell says, I don't care, because the alternative is I die anyway. So I might as well do whatever is possible to try to save myself, even if it means I'm a mutated mess afterward. Better alive and hurt than dead. So that is the SOS repair. Now, here's an interesting connection. Lex-A is going to repress these DNA repair genes. Lex-A also binds to really any DNA. It doesn't necessarily discriminate between bacterial DNA or viral DNA. So when we have a prophage during the lysogenic cycle, so a virus has come into the cell, taken its genome, and integrated its genome into the host chromosome, and now it is now a prophage. Lex-A won't be able to tell the difference between chromosomes that genes that came from the bacteria and viruses. So it will also bind to any of the genes of the prophage that trigger the lytic cycle. Remember, once the DNA of the prophage has been integrated into the chromosome, it can stay there, and it can stay there forever unless it recognizes that there's extensive DNA damage going on in the cell. And in those instances, it wants to protect its own DNA, so it will trigger the lytic cycle where it will start to make a bunch of baby viruses, and those baby viruses will burst out of the cell. So, Lex-A during normal, healthy DNA conditions is bound to these proteins of the prophage that otherwise would make the prophage convert into the lytic cycle. So it's keeping the prophage integrated into the DNA, and it's keeping it in the lysogenic pathway. But when we have this extensive DNA damage, Rec-A will start to break down Lex-A. And now these genes on our prophage are going to be turned on, and they are going to trigger a transfer from the lysogenic to the lytic cycle. And the cell will start to make a bunch of proteins for the viruses will assemble, and they will burst out of the cell. So this only happens when the host cell is under extreme harsh circumstances and it may not survive. Kind of cool that that connection is made, that the viruses are using a component that helps the cell to also help itself. Now, here's just a summary of all the different types of DNA repair. We are not discussing a couple of these, so just keep that in mind. You don't need to know those for the exam. Now, we're going to transfer into gene transfer mechanisms. When we are looking at bacteria and archaea, it's very hard sometimes to say you're part of species A and you are part of species B. Oftentimes, even using DNA, it's hard to tell them apart because they do a lot of gene transfer. They will give genetic elements to each other. And so we will see genes in some species that originated in completely different species or even completely different domains of life. So even if we are using DNA sequencing, there's a lot of mess when it comes to identifying bacteria and archaea. So taking up foreign DNA can be a good thing for the cell. It can be a useful alternative for food because, in the end, bacteria, even us, we can use DNA as a food source. In humans, we don't do it because it will cause a high buildup of uric acid in our body, which is quite dangerous for us and can lead to gout and things like that. But bacteria can definitely use it as an alternative food source. They can use external DNA to repair some of their own damaged DNA, or it can drive evolution. They might take on new traits that they didn't have before. Now, we have to talk about horizontal versus vertical gene transfer before we go any further. Vertical gene transfer is what you normally think of when you think of genetic inheritance. This is through sexual reproduction. One parent will mate with parent two, and they will have offspring, and the offspring are considered vertical transmissions of their DNA. Horizontal gene transfer is a little different. Rather than mom and dad mixing their DNA to make you, this is saying your mom goes to your dad and says, "You know, honey, I've always loved your blue eyes. I hate my brown eyes. Can you please give me your gene for your blue eyes?" And dad says, "Of course, honey, here you go." Mom takes that DNA from dad, sleeps on it, wakes up the next morning with blue eyes, just like dad's. So that is horizontal gene transfer, the transfer of genetic material from one organism to another to create genetic diversity without sexual reproduction. Now, one of these methods of horizontal gene transfer is conjugation. This is literally taking some DNA from cell 1 and transferring it over to cell 2, and it's going to be initiated by a special type 4 secretion system that we call the sex pilus. And this is going to protrude from the donor cell, it's going to attach to a recipient cell, and it creates a bridge. It actually connects the cytoplasm of both cells so the DNA can transfer between the two. So let's jump into this a bit. Conjugation requires a special plasmid. Um, This plasmid is called the fertility factor, or F factor, sometimes also called the F plasmid, and it's going to contain all the genes required to form that sex pilus, that type 4 secretion system, and it contains all the genes needed to export DNA. And those are often found in this TRA region of the plasmid, the transfer region. Now, if you look at this particular or plasmid, excuse me, there are two origins of replication, ORI V and ORI T. Now, this plasmid can just exist in a cell without conjugation occurring, and that cell might go through rounds of replication. And in that case, if the cell is not conjugating and the plasmid needs to copy itself, it copies itself starting with ORI V, origin V. So this is V for vegetative or non-conjugating cells, normally reproducing cells. And it will start here and it will reproduce the chromosome in that way. Now, ORI T is used during transfer. The T is for transfer. This is when we are going to initiate plasmid transfer. That way we initially start to transfer the genes for the TRA region. And those are the most important to get across. Now, excuse me, conjugation is going to begin when there is contact between a donor cell and a recipient cell. The donor cell is going to be called the F plus cell. The recipient is the F minus cell. Plus meaning it has the fertility factor, the F factor plasmid. Minus meaning it does not have this plasmid. We do not want a cell to to waste all this energy and all this time transferring a plasmid that the other cell already has a copy of. So because of this, we can only transfer the F plasmid to cells that do not have it already. So let's continue to explore conjugation. Now, as I said, it begins with the forming of a mated pair between an F plus and an F minus cell. Now, part of the way the cell can tell if a cell is F plus or F minus to initiate conjugation is that on the chromosome are genes that create these fimbriae or these projections on the surface of the cell. If the fimbriae exist because the cell has the F plasmid, then the sex pilus can't attach to that cell. It can't get past the fimbriae to attach and initiate conjugation. So that's the way that they can tell who is a recipient and who is not. Now, um, we connect, and immediately biogenesis of the sex pilus begins. And from here, what happens is the sex pilus is formed and reaches out to the F minus cell, and then it starts to contract, and it will pull the recipient cell closer and closer to the donor cell until they are literally touching each other, and we can open up a bridge between the two where they can share a cytoplasm. So it waits until it has made contact. It knows this because there's suddenly tension in the pilus, and then it will start to pull them together. Now, the F factor plasmid is going to receive then a single stranded nick at ori T and start to move across this one single strand. The F prime end is going to start moving into the F minus cell. And we call that through the bridge created by the sex pilus. DNA polymerase 3 is going to replicate the F plasmid in the F plus cell in a manner that is similar to rolling circle replication for how plasmids replicate themselves. And then once the DNA is completely transferred and it is a single stranded DNA in the recipient cell, it will also then just copy the DNA, and so that way it restores it back to being double stranded in the recipient cell as well. Now, the F minus cell is considered an F plus cell because it now contains the F factor plasmid. And only genes on the F factor plasmid get transferred. None of the chromosomal genes are being transferred during this time. So you can see the chromosome on the side here is untouched. Only that F plasmid is being transferred over. But life is not necessarily that clean. And sometimes the F factor can randomly integrate into the chromosome. There will be homologous sequences, so sequences on the F factor that are the same as sequences on the chromosome. And whenever that happens, we can have an event called homologous recombination, which is basically like crossing over during meiosis. And we end up inserting the whole plasmid into the chromosome itself. The chromosome is the dark gray here, as you can see here. And now we can see that there is ori V, ori T, and the whole TRA region all embedded within the chromosome itself. Now, this cell is going to be called an HFR, or a high frequency recombination strain. And this cell is going to be capable of transferring parts of the chromosome into the recipient cell when it initiates conjugation, because it can still initiate conjugation because it has the F plus plasmid. The plasmid is just now part of the chromosome. So the genes are going to start being transferred at the ori T, and they're going to start being transferred this way. But because there is no end, there's no terminator sequence basically until this portion here. The cell is going to keep saying, well, we're just going to keep copying then because the plasmid's not done yet. And so If the cell were to copy the entire chromosome and the F plasmid altogether, that would take about 100 minutes to transfer. Now, cells don't really wait that long. They're like, it should have transferred by now. We're going to break the sex pilus and move on with our lives. And so they're going to terminate it at some point down into the chromosome. So, which genes get transferred over and how many of them do just depend on when this whole process is terminated. But we've been able to use this in historical context to map genes on chromosomes before we were sequencing them because we know where the ORET is. So if it starts to conjugate and move over the chromosome, if we interrupt it after 5 minutes and gene A and B are transferred over, we know A and B are close to where that plasmid was. Um, if we see lots of copies of A and B being transferred over, then we know that they came over quickly or closer to the F plasmid. But if we allow it to go 10 minutes now, we may start seeing gene Z or gene M. And we'll see those less often than genes A and B because they're further down on the chromosome. So the frequency at which these genes get transferred over, we'll see the ones that are closer get transferred over more frequently, and how long it takes a gene to even show up. Gene A and B might show up in 5 minutes, whereas gene M might wait until 20 minutes. Gene Z might not even transfer over until minute 100. So we are able to get a crude rudimentary map of where all these genes were located. Now, um, when this happens, as I said, it doesn't usually transfer the entire chromosome because that takes too long. So we will end up with a small portion of the chromosome that got transferred over. If there's any homology between the small piece and the chromosome itself, you might have an event where they recombine, and you have a recombination event where now the donated chromosome gets switched out for the original chromosome of the recipient cell. And then either this extra portion will degrade or it might be used as a playground for evolution, because when we have two copies of the same gene in a bacterial cell, the bacteria will allow that second gene to incur extra mutations and basically allow mutations and evolution to take its course there to give the cell new abilities that it didn't have before. So this is just a little bit more detail about this. Because this extra piece is linear, it most likely gets destroyed afterwards unless it has a recombination event. And the new strain is technically not going to be F positive because the second half of the F plus plasmid is still in the original cell, the donor cell, the HFR. And unless the entire chromosome and the entire plasmid get moved over, the recipient will not become an F plus. So here is just a video about that. I highly encourage you to watch that. The HFR the F factor can leave the chromosome as well. It happens about the same frequency as the F plasmid joining the chromosome. The same percentage likelihood is possible for it to leave. So this can result in gene shuffling. We now can have these F prime factor, or F prime plasmids, where the F factor carries away chromosomal genes. When it pulls out, it takes too much DNA with it. And that can result in gene duplication when this F prime plasmid gets transferred into a new cell. There's also transduction. This is another method of horizontal gene transfer. And this is the process where the bacteriophage will carry host DNA away from one cell and into the other. And this is usually an accident. The phage is not doing this on purpose. And there's two types that we are going to discuss. The first is generalized transduction. This is not specific. Any piece of DNA from the donor cell can accidentally get packaged up and get sent over. So it's not necessarily where we can predict which DNA is going to move over. During the lytic cycle, when the phage DNA is injected into the bacterial cell, there's also phage enzymes that come in and cut up the viral DNA into smaller pieces. When that happens, these enzymes don't just cut phage DNA, they will often indiscriminately cut DNA, and sometimes they end up cutting the host chromosome. Now, the host chromosome is in small pieces as well. And as the polymerases are making a bunch of DNA, they're making the polymerases are, copying the DNA. They're making new proteins. They're creating all of the information needed to make new viruses. Well, as these new viruses are assembling, as their genome is getting packaged inside the capsid, sometimes they accidentally take up DNA from the bacterial chromosome as well. So instead of packaging up just viral DNA, they accidentally package up bacterial chromosome DNA. Then they burst out of the cell, and when they go and infect a new cell, they might accidentally insert this new DNA that does not belong to the phage but to the bacteria. When this happens, we can end up with a recombination event, because if this piece of DNA is homologous to the chromosome and the DNA might just swap places. And this can introduce new genes into bacteria and is often used in the lab for us to introduce the gene that we want, often a mutated gene. Then there is specialized transduction. So this is going to occur with a lysogenic phage rather than a lytic phage. So in this case, the the DNA of the phage gets integrated into the host chromosome, and when it pulls out of the chromosome, it may accidentally take some chromosomal genes with it, just like the HFR. When it pulls out of the chromosome, it doesn't necessarily just take itself. It may take extra genes that are close to the insertion point as well. So this is why it's more specialized rather than generalized, because it only transfers the genes that are closely linked to the insertion site of the phage. It's not randomly taking from anywhere on the chromosome. So in this case, the host recombinases remove the phage DNA. They accidentally cut out some chromosomal genes, and those move away and get packaged into new phage and get transferred over to a new cell. Then there is transformation. We talked about this with Griffith's experiments with um, the bacteria that were harmful, and then he heat killed them and they did not kill the mouse. But when he mixed the dead heat killed harmful bacteria with harmless bacteria, the harmless bacteria took up the harmful DNA from the bacteria that were the killer bacteria, took them up into themselves, and then became harmful killer bacteria. um, I know those aren't quite uh, exactly specialized terms there, but that was just a quick breakdown of it. So, transformation is where the cell will import DNA that's just freely found in the environment into the bacterial or the archaeal cell. Not all cells can do this. Only competent cells can do this. So they have to have the ability to take up the DNA. And most bacterial cells are not naturally competent. Now, there are inherent dangers to taking up DNA from the environment and other bacteria. It could be dangerous. You might get a gene that is harmful to you. You might get a viral gene that ends up killing you. So, it is important that bacteria have safety mechanisms to check and verify the incoming DNA. And this can include restriction endonucleases that will recognize certain sequences in DNA and destroy it, or protective methylation, where we say that the bacteria methylate their DNA, and that's how they tell a difference between their DNA and viral DNA, and then they can target the viral DNA for destruction. So let's talk about restriction endonucleases briefly. These are enzymes that cut DNA, and the region that they cut is a palindromic sequence. So they recognize sequences that are read the same direction forward and backward. So same in the 5 prime as 5 prime to 3 prime as in the 3 prime to 5 prime direction. Now, when DNA is methylated, it is protected from these restriction enzymes. It cannot be cut by these endonucleases. So it requires unmethylated DNA, which also helps the cell target that foreign incoming viral DNA rather than its own. There are also transposons. These are mobile genetic elements or transposable elements. They're basically sequences of DNA that can cut themselves out and move to another part of the DNA. They cannot exist outside of a larger DNA molecule. So they're not just found in the environment and then will suddenly jump into your cells. They exist in DNA. They can copy themselves into some other place on the gene, or they can cut themselves out and move to another place in the chromosome. And these are found in all forms of life. This is not just a bacteria or archaea thing. Eukaryotes have this too. We use them in research all the time because they can be used in all domains of life. Now, an insertion sequence is going to be the simplest type of transposable element. It has the transposase gene, so the gene that codes for the protein that helps it do the jumping, and it has short inverted repeat sequences on either end because that's where it knows where to cut. The enzyme knows where to cut. And these are going to then cut themselves out and move somewhere else. Now, when these transposable elements transpose or move, they do it by two different mechanisms: replicative and non replicative transposition. With replicative transposition, we are replicating the transposon first. So here we see the transposon in between genes A and B, and it has a target sequence in between genes C and D. For replicative, it will copy itself and then insert the copy in between C and D. So in the end, the transposon is found between A and B and C and D. Non-replicative, it cuts itself out of A and B and jumps and inserts itself in between C and D. This one is more inclined to induce mutations because we are jumping out of the DNA, which could lead to DNA being lost or taken up with the transposon. Maybe the transposon inserts itself in between a gene. It just depends. Now, moving away from gene transfer mechanisms, we're going to close off with a brief discussion on how genomes evolve. Now, remember that microbes are living from one extreme condition to another, basically one starvation event to the next, one toxic environment to the next. They have to be able to be quickly adaptable. So, their genomes have to allow them to be very dynamic so that they can continually evolve to their changing environments. And genomes usually evolve through gain and loss of genes, as well as mutations, of course. But there's two basic ones that we're going to discuss today, and horizontal gene transfer and duplication are those two. We've discussed horizontal gene transfer ad nauseum at this point. You can see how that can introduce new genes or new gene fragments or cause recombination events that maybe bring different versions of the gene, different alleles in, basically. So that is discussed already. Let's discuss this homology, duplications, and divergence, though. So, gene duplication is probably the most important mechanism to generate new genes and new biochemical processes. So, we copy a gene, and now this duplication is not needed for cell survival. The cell doesn't need to make sure that this duplicated gene is replicated faithfully. It doesn't need to be super strict on making sure that mutations are fixed immediately. So, through divergent evolution, mutations will build up, and eventually, this gene may take on a completely different role that didn't even necessarily relate to the gene that it came from before. This is very common in plants. It's plants tend to be like tetraploid or even further ploidy, where they have, not just four, but multiple extra copies of their genes, and they allow many of these extra copies to, have evolution act on them. With bacteria, they only have one copy of their chromosome. So any duplication event, they only need that one copy. Any secondary one that they get to either kick it out, or they will allow evolution to take its course and let mutations run rampant in that extra gene copy. But sometimes it is not useful to keep that secondary copy of the chromosome. It's not necessary for the cell, and in fact, in certain situations, it might be harmful for the cell. So, genome reduction is when the cell will large scale lose genes through evolution. If you have a very large genome, it's going to take more effort to maintain, because in the end, you still do have to copy these genes. You do have to do some maintenance of these genes. You will have to transcribe these genes. So, if you have a gene in your genome that is completely unnecessary, the actual act of needing to maintain this unnecessary gene might make you less fit than the other cells around you who don't have these extra gene copies or who don't have these large genomes. If you are using up extra energy just to maintain a gene you don't need, you can't use that energy to fight off infection, to duplicate yourself, to survive in harsh environmental conditions. So it would be in your best interest to get rid of those genes. If you happen to get rid of them, you will survive better. So, some examples of that: there is a Shigella species. These are pathogens that are highly, highly related to E. coli. In fact, a lot of recent discussion amongst scientists and experiments into it have basically found that E. coli and Shigella are basically the same species. Shigella is like a subspecies of E. coli. So, in pathogenic or harmful Shigella species, they have chromosomal black holes. So, these are areas where, well, E. coli has genes here, but they don't. So, they lost those genes over time because for some reason those ones weren't particularly needed by Shigella. The Mycobacterium species, like Mycobacterium leprae, the acid fast bacteria, the one with all the mycolic acids in its cell wall, particularly one that causes leprosy. Its genome has many non functional pseudogenes. So, kind of imagine this as a halfway point rather than just, having these black holes. We still have the gene there, but if you look very closely at these pseudogenes, first glance, oh yeah, you think that encodes a protein. It looks like it encodes an enzyme, but then deeper looks into it find that that protein or enzyme is non-functional, or the gene itself is non-functional. It's just there, but it's not working. The cell isn't allowing it to expend resources to turn it into something functional. Now, this is very common in bacteria like Mycobacterium leprae. All of the Mycobacterium species are intracellular pathogens, meaning that they live inside your cells. They don't live outside of your cells. When they live inside your cells, there's many genes that they don't need to keep anymore, because why am I going to keep an S layer if I'm in this really nice cozy environment all the time? Why am I going to maintain these housekeeping genes when the cell takes care of all that stuff for me? It breaks down the food for me, and I get the food pieces that just float into my cell. Why would the cell maintain, um, a lot of stress genes like acid stress or things like that when it's inside a nice cell that maintains its own homeostasis? So, in these obligate intracellular pathogens, we often see them lose big portions of their genome or have these pseudogenes because all of this genetic material is now no longer needed because they're using the host machinery instead. And then one last thing we're going to talk about: horizontal gene transfer causes some very interesting things to occur. particularly genomic islands. So if we look at any given species of their DNA, take E. coli, it will have some percentage of CG content. Then you take maybe salmonella, and it will have a slightly different percentage of CG content. And we can kind of track what species is what based on its CG content in its DNA, how many C's and G's it has, because those ones, remember, form 3 hydrogen bonds instead of 2, so they're a bit more stable and can survive in extreme environments a bit better. So we see a lot of extremophiles with very high CG content in their DNA. Prokaryotes in general have higher CG content than eukaryotes do. So what we can do is take a look at a map of the CG content of a cell. And we will say that, okay, the average CG content is about 53, 55% for this cell. But suddenly, this one region of the chromosome, the average GC content dropped down to like 30. So, why is this one so different from the rest of the cell? Well, horizontal gene transfer. It probably got that set of genes through a conjugation event, through a transduction event, a transformation event, some event where it got these DNA sequences from a species other than itself. And so we end up calling these genomic islands because they're islands where the DNA does not belong necessarily. And so we have a couple different types of genomic islands. One common one that we're probably going to talk about later in the semester is a pathogenicity island. This is where that region is going to encode a lot of genes that make the cell more virulent and more able to cause disease. Symbiosis islands, this particular group of genes will allow it to exist as a symbiont. And fitness islands are probably ones, or are ones that will help the cell survive in alternative conditions. So if we want to have a higher level of fitness, we want to be able to survive in high stress, high salt, high acid, cold, all these different stressful environments. And that is it. So make sure you study these study questions. Let me know if any of them do not make sense, and I would be happy to help you with that. Otherwise, I hope you have a great day and talk to you soon.